Hi friends, today we are going to solve Rpsc AARO Botany paper 2, which the exam was in 2022 in August. So we will solve the botany part. So question number 1 is, first is the GK part, which we are not discussing, we are only discussing the botany part. So botany part starts from question number 31. So question is, if n inbred lines are crossed together, number of double crosses can be calculated by the formula. So number of double crosses are calculated by the formula n minus 1, n minus 2, n minus 3 by 8. When it is double cross, it will be divided by 8. Okay, so answer 3 is correct. Next is, plant with gametic chromosome number are termed as which of the following? If it is gametic, it is haploid. If it is sporophytic, it is diploid. So answer is haploid. Next is, what will be the sequence of damage caused by various organisms? So sequence of damage, the most damage is by fungi, then bacteria, then viruses, then nematodes. Okay, so the sequence of the damage most is by fungi. Answer 2 is correct. Next is, chromosome number of cowpea is. So chromosome number of cowpea is 2n is equals to 22. Next is, nucleus was first discovered by. So nucleus was discovered by Robert Brown. Next is, which one of the following is correct for mutations? So mutations is caused by heritable changes in genetic information. Okay. Other options are caused by genetic recombination, caused by faulty transcription of the genetic code, usually beneficial to the development of the individuals in which they occur. So all the options are like you can say partially correct, but they are not appropriate. Answer 2 is appropriate, where it can be heritable or it can when it is due to physical mutagens in the single generation. So most of the time it is heritable. Okay. Next is, which one of the following statement is not correct? So wild forms are wild species from which crop species were directly derived. So wild forms are not wild species. Okay. Next is, wild forms are easily crossable. They are not easily crossable. Okay. Next is, wild relatives include all of the species which are related to the crop species by descent during evolution. So wild relatives, it does not include all of the species, okay, which are related to crop species. Next is, wild relatives are much easier to hybridize with crops than are wild forms. So wild forms are not easier to hybridize, instead wild relatives are easier to hybridize. Next question is, use the following events to answer the question. So what is the question? The correct order of events required for protein synthesis is. So when you will read translation process, the fundamentals, you will find that first of all what will happen? First of all, DNA segment will open up. Okay, replication will occur, then transcription will occur, then translation will occur. So first of all, number 2 will be correct, which is DNA segment will open up. Then mRNA will be formed, which is transcription. Then mRNA attaches to the ribosome, here where translation starts. Okay. Then tRNA carries amino acids to the mRNA. tRNA enzyme will carry the amino acid to the mRNA. Peptide bond will be formed. And next is, amino acid forms peptide bond. Okay, there is a transfer of peptide amino acid from one site to another. If you read the topic in detail, you will understand. So here answer is 3. Next is, lead center for DUS testing of rice is located at. So DUS testing of rice is located at Hyderabad Indian Institute of Rice Research, IIRR. Okay. Next is, the germplasm is the sum total of hereditary material in a species. It is defined by. So germplasm is defined by the scientist named Allard. Okay. Germplasm is defined by the scientist named Allard. Okay. Next is, if a somatic chromosome number is 2n is equals to 20, the number of chromatids present at the end of the prophase of mitosis is. So at the end of prophase of mitosis, it will be doubled, which is 40. Next is, in base collection, germplasm can be stored for. So base collection, germplasm could be stored for about 100 years. Okay, answer 4 is correct. Next is which is not correct about free radicals. So free radicals, they produce genetic effect, they have a very short life, they have unpaired electron which makes them more reactive. But they are not electrically charged. Okay. Next is a nucleotide is made up of which of the following chemical components? So nucleotide is made up of nitrogenous base, pentose phosphate group and pentose sugar. Next is which one of the following combination is correct? So here selection of superior types in M1, prevention of outcrossing in M1, prevention of mechanical mixture in M1, progeny rose in M1. Here mechanical outcrossing should not be there in M1 and mechanical mixture should not be there in M1. Okay, B and C are correct. Next is among the given hydrogels, which one is the most preferred one? So sodium alginate is the most preferred hydrogel. Next is size of foundation seed tag is 15 centimeter into 7.5 centimeter. Okay, it is 15 centimeter into 7.5 centimeter. Do you know what is the size of herbarium? If you know the size of herbarium, can you write it in the comment box? So one homework for you, write the size of herbarium in the comment box. Next is the DNA product of transcription is RNA. Okay. Also another question, do you know like which RNA is formed at which RNA is formed in the highest amount after transcription? Okay, there are three kinds of RNA: mRNA, tRNA, rRNA. Which one is formed in the highest amount? That you have to write. Next question is which one of the following statement are not correct about mutation? So which one of the following statement are not correct about mutation? If mutation occurs in a germinal cell before differentiation of gametes, it would influence several gametes and thus will influence all individuals derived from affected gametes. So if the mutation is in the gametic cells, okay, and differentiation has not occurred yet, okay, many cells have not formed yet, in that case that one cell will influence many other cells, okay, which are derived from that gamete. So next is if mutation occurs in a gamete or a zygote, a single individual will carry the mutation. So later on, if the mutation occurs, the single individual will carry the individual whoever is formed from that zygote will carry the mutation. Next is if mutation occurs in a cell after the zygote has undergone one or more division, only a part will show the mutant character. Okay, so if zygote have undergone differentiation and some cells of the zygote only shows mutation, that means that those cells will only show mutation, and those cells, whichever organ those cells are forming, that only will show the mutation. Okay, whole body will not show the mutation. Number D, if mutation occurs in a gamete or a zygote, all individuals will carry the mutation. So if the mutation is in the zygote, all will carry the mutation. So see, if it is in the zygote, depends which part. If it is in the whole zygote, then there are the chances that all the organisms, all the next generation will have the disease but have the mutation. But depends, like this is a little dicey. It's like all the individual may carry the mutation if it is in the zygote. Okay, so this is something which is partly correct, so you can check over this, and if you have any comment on this, you can write it in the comment box. Next is match the following. So dominant epistasis is 12:3:1, recessive is 9:3:4, duplicate dominance is 15:1, and duplicate recessive is 9:7. Answer 4 is correct. Next is a gene that influences more than one trait is called pleiotropic gene. Next is in primary trisomic, the extra chromosome is. So in primary trisomic, the extra chromosome is normal. Next is, we all know alternative form of a gene is allele. Next is cell remain plasmolized even after thawing is known as. So when cell remain plasmolized even after thawing, it is known as frost plasmolysis. Next is a somaclonal variant of linseed released as a variety for commercial cultivation. It is called andro. Okay, andro variety of linseed. Next is Brassica junctia is evolved from a cross between. It is evolved from a cross between Rapa and Nigra. Brassica rapa and Brassica nigra. So Brassica junctia having AA and BB genome. So AA genome is coming from one species and BB genome is coming from one species. So that is why Brassica junctia have AABB genome. Okay, it is polyploid species. Okay, next is an autotriploid contains 3 copies of one genome. Okay, next is the plant whose pulp is used in paper industry. We all know bamboo pulp is used in paper industry. It is bambusa. Next is select one of the following which is not related to maintenance breeding. So plant to progeny method is related to maintenance breeding. Positive mass selection and negative mass selection is related to maintenance breeding, but back crossing is not related to maintenance breeding. Next is Pigeon P hybrid ICPH8 was developed from a cross between. So Pigeon P hybrid is developed from a cross between MS Prabhat and ICPL 161. Okay, MS Prabhat and ICPL 161. Next is in the Helminthosporium sugarcane system, which one of the following combination will show susceptible response? Okay, which combination will show susceptible response? So when in the susceptible response, what happens? The pathogen should be resistant. The host should be resistant and pathogen should be virulent. Okay, so if pathogen is virulent, in that case there will be susceptible response, and you can see the gene for host is RR. Small r is what? It is susceptible. Capital R is what? It is resistant. But here if the resistant gene is not there with the host, but pathogen in the recessive condition, the PP gene in the recessive condition, it is virulent. So even if Helminthosporium is not mentioned, you could have answered this question. If there is a virulent gene dominant and the host gene is recessive, in that case there will be susceptible response. Next is match the information of a group A with group B. Okay, so group A additive variance. Additive variance is fixable. Okay, when there is additive variance, it is fixable. When there is dominant variance, opposite will be there, it is non-fixable. When it is polygene, okay, when it is polygene, it is like given by Mather 1941. And what is epistasis? Epistasis is interallelic interaction. So additive variation is what? Fixable. Dominance variation is non-fixable. Polygene by Mather 1941. Epistasis by interallelic interaction. So which one is correct? Answer D is correct. Next is, if the code for an amino acid is AGC on the DNA molecule, the anticodon on the tRNA would be. So what will be the anticodon on tRNA? What will be the anticodon on tRNA? Okay, if the code of an amino acid is AGC on the DNA molecule, so the mRNA would be changed. Okay, but the anticodon on the tRNA will be complementary to the mRNA. Okay, it will be not be complementary to the DNA. Okay, so it will be what? It will be AGC only on DNA. Okay, so remember whether it's given DNA or whether it is given tRNA, or sorry, whether it is giving mRNA. Okay, next is who can issue the breeder tag? Who can issue the breeder tag? Breeder tag could be issued only by breeder. Next is bioinformatics include which of the following activities? So bioinformatics include preparation of database, development of development of hardware is not the work of bioinformatics. So it is preparation of database, compiling of genome sequence, assigning function to the identified gene. So A, B and D. Hardwares do not like bioinformaticians do not develop the hardware. Next is, which one of the following statement is not correct about DNA replication? So, DNA replication always proceeds in the 5' to 3' direction, true. The presence of replication fork in E. coli chromosome was first demonstrated by Cairns. It is also true. Next is, both strands of DNA replicate continuously. It is wrong. One strand replicates discontinuously, that is Okazaki, the lagging strand. Next is, DNA replication begins with a synthesis of small primer. This is also correct. Moving forward, in case of recurrent selection for SCA, the tester that is used has, okay, in case of recurrent selection of SCA, the tester that is used is narrow genetic base for recurrent selection. Next is the possible phenotypes with 4 different alleles of a multiple allelic series. Okay, the possible phenotype with 4 different alleles. When there are 4 different alleles of a multiple allelic series, then what will be the possible phenotype? Phenotype will be 4. Okay, genotype can be multiple, but phenotype can be 4. Okay. Next is, which one of the following species crosses give the highest hybrid heterosis in rice? So heterosis is what? Heterosis is when the F1 generation is better than the parental generation. Okay. So which species of rice give highest hybrid heterosis in case of rice? So indica and japonica, when they are crossed, it gives the highest hybrid heterosis. Next one is, which one of the following crop is the most sensitive to salt stress? So beans are the most sensitive to salt stress. Next is, which applies to Compton scattering? So what applies to Compton scattering? Compton scattering is a high energy photon spends a part of its energy to kick away an electron from an atom. It is actually kicking away of electron from an atom. Okay, so photon which is spending a part of its energy to kick away an electron means light. What will happen? The photons present in the light, it will spend some part of its energy to kick away the electron. It will move away the electron from an atom. This is Compton scattering. Okay. Next is, match the column A with column B and pick the correct option given below. Excuse me. So particulate radiation is thermal neutron. Okay, having thermal neutron. Next is non-particulate radiation is X-ray. Next is non-ionizing radiation. Non-ionizing radiation is for non-ionizing radiation, what is the example? It is UV rays. Non-ionizing is UV rays, and base analog is 5-chromouracil. So particulate radiation, if I mark, particulate radiation is by thermal neutrons, non-particulate is by X-ray, non-ionizing is UV, base analog is 5-chlorouracil, bromouracil, and so on. So answer here is C. Next is, match the following again. So you have to name the scientist, whoever have discovered this. Okay, so metroglyph analysis is by the scientist Anderson. Metroglyph analysis is by the scientist Anderson. Okay, path analysis is by the scientist named Wright. Okay, path analysis is by Wright. Next is triallel and quadriallel. Triallel and quadriallel is by Rawlings and Cockerham, and partial diallel is by Kempthorne and Carnot. So they are very difficult to remember all the scientists' name. I would suggest you go, if you remember it's okay, otherwise don't do these type of questions. So answer here is B. Next if the ratio of X by A is 1 in Drosophila, the individual is. So there is a different ratio which can determine whether it is male, super male, a female, super female. So if the ratio is 1, in that case it is female. Okay. Next is the first report on heterosis was reported by. First report on heterosis was reported by the scientist named Coyle Reuter in 1763. Next is Ricinus communis is a. Ricinus communis is a very important, okay, it's a very important shrub, have lots of uses, and it's a very common topic for the examiners to ask the questions. Okay, please don't forget like the floral formula, the family of Ricinus communis, everything is very important. Next is match the following. So here hybrid is what? Hybrid is hybrid is F1 generation grown commercially. Okay, this is hybrid, which is F1 generation grown commercially. It could not fit in one page, so I am just showing you F1 generation grown commercially. Okay, pure line is what? Pure line is progenies of single self fertilized plant. Okay, pure line is there is no mixture of genetic information. Okay, pure line is when one generation is grown for multiple times, okay, which is single self fertilized for multiple times. Okay, this is pure line. Next is composite. What is composite? Composite is advanced random mated generation of crosses. Okay, composite is when there is random mating and generations formed from there. Okay, number 1. Synthetic is what? Synthetic is maintained through open pollination and component lines have been tested for GCA. Okay, so synthetic is when they are open pollination. Pure line is, say for example, when single generation is like produced by self fertilization. Hybrid is what? F1 generation when grown, it is hybrid. Then pure line already told. Composite is when there is random mating, generation of crosses. Synthetic is when there is open pollination and the component lines have been tested for GCA. So answer is 3. Next is again very important question of the examiner, a very common question and favorite question of the examiner. Flavor saver is a transgenic variety of. So flavor saver is the transgenic variety of tomato. Next is the mRNA which are coded by one cistron. So when it is one cistron, it is monocistronic as the name says. Next is select the wrong statement among the following. So select the wrong statement among the following is heterosis can be fixed by vegetative propagation. Absolutely correct. Yes. Heterosis can be fixed by apomixis. True. Heterosis can be fixed by balanced lethal system. True. Heterosis can be fixed by inbreeding. So this is absolutely incorrect. Heterosis cannot be fixed by inbreeding. Next is which one of the following statement regarding cytoplasmic inheritance is true? Okay, cytoplasmic, which one is correct? So equal contribution, correct, it's wrong. It is more by female. Linkage relationship operates in plasmogen, no linkage relationship. Equal distribution, no. Reciprocal differences exist. So it is absolutely correct. Reciprocal difference exists in case of cytoplasmic inheritance. Next is so many match the column came this time and they are very scoring. Okay, so this you memorize by heart. Which chromosomal aberration causes which syndrome? Okay, so Down syndrome is by trisomy? Tell me please. Down syndrome is by trisomy 21. Okay, Down syndrome is by trisomy 21. Okay, Patau syndrome is by, tell me, Patau syndrome is by monosomic. Edward syndrome is trisomic 18. Turner syndrome is, sorry, I did wrong. So Down syndrome is trisomy 21. Okay, Patau syndrome is trisomy 13. Edward syndrome is 18. Turner syndrome is monosomic, which is XO. Okay, so answer here is number A. Next question is, the PCR technique was developed by, we all know, it's another favorite question of examiner, by Carey Mullis in 1985. Next is, which one of the following is not membranous organelle? So non-membranous organelle are ribosome, again another favorite question I'm circling. Adenine and guanine, another favorite question. There are so many favorite questions of examiners which you should know. Okay, should know. So adenine and guanine are what? They are purine bases. Next is improved Pusa Basmati 1, Pusa 1460, the first variety in India developed through MAS, okay, marker assisted selection, has been improved by transferring which of the following? So which of the following have been has been improved by transferring and forming Pusa Basmati? So it is for disease resistance. Okay, this Pusa Basmati 1 is for disease, more disease resistant character have been improved. 87. Next is assertion. Leaf rolling in species like wheat and sorghum predicts turgor maintenance. This is because these species show relatively better osmotic adjustment than does rice. So which one is correct and which one is not? Okay, so leaf rolling is seen in wheat and sorghum, which shows that they have turgor maintenance like character, and this is because these species, and because they have leaf rolling ability, they can show better osmotic adjustment than does rice. So this is absolutely correct, and R is the correct like explanation of A. Next is low magnitude of genotype into environment interaction includes. So low magnitude of genotype environment interaction includes what? It includes high buffering capacity. Next is RFLP. RFLP is what? It is hybridization based marker. Okay, codominant hybridization based. Next is Cicer species, annual small flowered species with firm erect to inclined or prostrate. Here it is asking which Cicer species is having firm erect to inclined or prostrate stem. It is monocicer. Next is pigeon pea. So another question we have done about pigeon pea. What was that? Can you remember what was the question we did for pigeon pea? It was that pigeon pea hybrid ICPH8. It was developed by a cross between Prabhat and ICPL 161. Okay, Prabhat and ICPL 161. Another question comes about pigeon pea. Pigeon pea chickpea. So ICPL 8 was there, right? ICPH 8 was developed by a cross between MS Prabhat and ICPL 161. Next question is, pigeon pea, chickpea, mung bean and cowpea originated in. So they all have been originated in Hindustan center of origin. Next is the plant family included under the series Unisexuales. So Unisexuales are included in the series U4BAC. Next is the germ plasm which is collected from foreign country is referred to as. So when germplasm is collected from foreign countries, it is referred to as exotic collection. Next is the genetic constitution of a normal Drosophila female is. So the normal Drosophila female is XX and the male is XO. Next is a deleted chromosome can be. So deleted chromosome can never revert to normal condition. Next is which of the following statement is not correct. So cytoplasm does not play a role in expression of heterosis. So this is not correct. Okay. Next is in sorghum, some pure lines varieties are almost as good as many commercial varieties. True. The superiority of modern maize hybrid is primarily due to their ability to perform well at high planting densities. True. Hybrids show 20% or more yield improvement over open pollinated varieties. This is also true. Next is TBG 39. TBG 39 is a variety of. So TBG 39 is a variety of groundnut. Next is first artificial hybrid of carnation and sweet Williams was made by. So it was made by the scientist named Thomas Fairchild. Okay, Thomas Fairchild, which is hybrid of carnation and sweet Williams. But we have also studied about Mather. You remember Mather scientist? We have also studied about him. Can you find it out and write it in the comment box what we have studied about the scientist Mather? I am giving a pause. You can wait and search. You can go back and search about the scientist Mather, because if you can relate it to the previous question, then only you can recall. Okay, so we have studied in the previous match the column where Mather scientist has invented the term polygene. Okay, has invented the term polygene in the year 1914. Okay, but here it is for carnation, it will be Thomas Fairchild. Next is biotype differentiation relates primarily to. So biotype differentiation relates primarily to insects. Next is if a variety is found suitable, it is released for commercial cultivation by. Who releases the variety for commercial cultivation? The Central Variety Release Committee and the State Variety Release Committee. Both has to agree. Okay, then only the variety will be released. Next is the most common gene pool used in breeding program. So most common gene pool used in breeding program is GP1. Next is transfer of some genes from one species into the genome of another species. It is called as introgression. Okay, gene from one species to another species, it is called as introgression. Next is choose the wrong statement of the following. So choose the wrong statement. Incompatible reaction of a gene found in biotrophic pathogens. So incompatible reaction occurs in biotrophic pathogens. True. Complete reaction of gene for gene found in facultative parasites. In facultative it is complete reaction. In biotropic it is incompatible reaction. Next is accumulation of phytoalexins at the site of infection is followed by cell death. It is also true. Next is vertifolia effect was due to variety carrying low of VR and high level of HR. So this is wrong. Okay. Next is the concept of dirty crop multi-line approach. Dirty crop multi-line approach is what? It is given by the scientist named Marshall. Dirty crop multi-line approach. Next is among the given auxin, which one is most frequently used in plant tissue culture? It is synthetic auxin 2,4-D. Next is real value of seed lot is determined by. So real value of seed lot, it is determined by purity percent into germination percent by 100. Okay. Next is gene bank for forage and fodder crop is situated in. So gene bank for forage and fodder crop, it is situated in Jhansi. Next is one gene, one hypothesis was given by the scientist Beadle and Tatum. Next is which one of the following generates gamma rays in the irradiated material. So what generated gamma rays? Neutrons. Next is which one of the following classification is a sexual system of classification? It is artificial system is a sexual system of classification. Next is addition of deletion of bases causes what kind of mutation? So it causes frame shift mutation. Next is headquarters of ICGEB is located at. So headquarters of ICGEB, it is located at Tri-Este. The first strategy used for molecular mapping was based on which of the following? So the first strategy used for molecular mapping is near isogenic lines. Which one of the following includes genetic engineering? So genetic engineering includes gene revolution. Certificate is not required for. So certificate is not required for nucleus seed and breeder seed. And who releases breeder variety? Breeder. Okay. So certificate is not required for nucleus seed and breeder seed, but it is required for foundation and certified seed. Next question is flag smut disease of wheat came into India from. So flag smut disease of wheat came into India from Australia. Next is which one of the following does not describe over-dominance hypothesis of heterosis. So which one of the following does not describe over-dominance hypothesis of heterosis? It is heterosis is due to heterozygosity. This is true. It is most applicable to maize. This is absolutely incorrect. How can it be most applicable to a particular crop? Next is inbreeding depression is due to homozygosity. No, it is kind of related to homozygosity only. This is also correct. It is exhibited by sickle anemia gene. So kind of irrelevant, but which of the following does not describe over-dominance hypothesis? It is true. So rest are something irrelevant. Next question is match the following. So ghost gene hypothesis. Ghost gene hypothesis is horizontal resistance. Ghost gene hypothesis is horizontal resistance. Then horizontal resistivity. Horizontal resistivity is polygenic control. Okay. Next is race specific. Race specific is vertical resistance. Hypersensitive response shows cell death. So here option A is correct. Okay. Race specific is vertical resistance. Hypersensitive response is cell death. Horizontal resistivity shows polygenic control. Ghost gene hypothesis shows horizontal resistance. Next is the right of an author, artist, publisher to retain ownership of works and produce or contract others to produce copies is called. So it is copyright. Okay, which is the right of the author. Okay, which is like the others cannot copy if he has like published that work. Okay. Next is the triple cross analysis involved. So triple cross analysis involves F1, F2, P1 and P2. Next is a genetic locus that serves as a recognition site for RNA polymerase. It is promoter. Next is the first transgenic crop plant produced is tobacco. Next is match the column. Endosperm, endosperm, definitive nucleus. Seed coat, integuments. Perisperm, nucellus. Seed ovule. So answer C is correct. Next is, which one of the following crops shows highest inbreeding depression? So highest inbreeding depression, which is due to homozygosity. Okay, cannot be reversed by heterosis. So it is carrot. Next is, the male sterile line TIFT 23A of Bajra was introduced from USA. So again match the column. Heterosis, hybrid vigor. Overdominance, superdominance. Luxuriance, pseudoheterosis. Useful heterosis, economic heterosis. Okay, so answer here would be A. Next is law of parallel variation. Law of parallel variation was given by scientist Vavilov. Next is, match the column A carefully and select the list given below. So match. Genome is the complete set of genetic material or genetic information. Proteome is the complete set of proteins. Genomics is the genome structure and function. So genomics and genome is complete set, but genomics is the genome structure and function. And proteomics is determination of proteome. So genome analysis is genomics, protein analysis is proteomics. Answer here is C. Barbara McClintock was awarded Nobel Prize in the year 1983 for. So the Nobel Prize have been awarded for transposable elements, discovery of ACDS, transposable elements. Next is, father of hybrid cotton is C.T. Patel. Next is, in plants, male sterility tends to increase. So male sterility tends to increase outcrossing. Next is, when the value of selection coefficient unit is S is equals to 1, survival rate will be. So when S is equals to 1, there will be no survival. When selection coefficient is 1, there will be no survival. Next is, in the cell, the chromosome are in the condition of maximum spiralization and are placed along the equatorial zone. So whenever there will be equatorial zone mentioned, it will be metaphase. Next is match. So again here wheat is 1:80. Okay, wheat is 1:80. Then black gram is 1:40. Okay, this is the seed multiplication ratio. Wheat is 1:80, black gram 1:40, linseed is 1:50. Okay, so wheat and black gram is totally opposite. Okay, linseed is the midway, 1:50, and half of the wheat is rice, which is 1:. I did wrong now. So wheat is 1:80. Okay, wheat is 1:80, black gram is 1:40, linseed is 1:50, and rice is again, okay, so wrong, wait. Wheat is 1:20. Okay, wheat is 1:20. Wheat is 1:20. Then wheat is 1:20. Then black gram is 1:40. Okay. Then linseed is 1:50 and rice is 1:80. Okay, so rice is having highest seed multiplication ratio. Wheat is less, 1:20 least. Linseed is 1:50 and black gram is 1:40. So answer here is C. Maximum seed lot size in maize. So maximum seed lot size in maize, it is 400 quintal. Next is, the performance of a synthetic variety is affected by. So the performance of the synthetic variety, it is affected by mean performance of F1 and number of parental lines. So all will affect the performance of synthetic variety. Okay, mean performance of F1 and the number of parental lines. So answer 4 would be correct. Next is most of the mutant chimera in clonal crops. So most of the mutant chimera in clonal crops are periclinal chimera. Next is DNA replication takes place during which phase of the cell cycle? Very important question for the examiner. It is S phase. Next is under Companies Act, SFCI was set up in the year. So it was set up in the year 1969. Next is canopy temperature is readily measured with. Canopy temperature is measured with infrared thermometer. Next is match the following. AVRDC Institute is located in Taiwan. Okay. Next is ICARDA, it is located in Syria. Next is WARDA, it is located in Liberia. IITA, it is located in Nigeria. Okay, so the correct answer would be C. So you need to revise all the questions again and again, and we will keep solving number of papers so that you can revise, watch the videos. Once you watch the video in 1x speed, then once you are done, you can watch the video by increasing the speed so that you can revise. And more you revise, the more familiar you will become with the questions, and that's the secret of qualifying any exam, be it agriculture exam, be it botany exam, be it any exam where the botany is a part of that. Okay, any teaching exams or any master exams for PhD exams or graduation exams. Okay, or any professional level examinations. So again, let's move forward. The largest number of varieties developed through hybridization between mutants belong to. Largest number of varieties developed through hybridization between mutants belongs to cereals. Next is crop improvement can utilize which of the following mutations? It can utilize vital mutation, which is important for the crop. Next is the full complement of RNA molecules produced by the genome. The full complement of RNA molecule, the whole RNA molecule, it is transcriptome. Next is the concept of gene pools was proposed by, it is proposed by the scientist named Harlan and Dewitt. Next is the range of correlation coefficient is, it is -1 to +1. Okay, next is again very important and favorite question for the examiner is DGO Wu gen is a variety belonging to which of the following species? It is belonging to the Japonica variety. Also we have done one question, Japonica and Indica shows like good F1 generation varieties. Okay, highest heterosis. Next is which of the following is used for ELISA? So for ELISA, what are used? For ELISA, antibody is used and antigen is used. Okay, B is correct. Next is first degree of statistics include, it includes mean. Next is population of cross-pollinated crop species are. So cross-pollinated crop species are heterozygous and heterogeneous. So here we end our 150 like total 120 questions because 30 questions was of GK. So this exam was in August 2022 and the question are very simple. Just what you need is you have to practice a lot. Okay, what is the key to success? Success is practicing. Okay, you have to practice a lot. okay, and we will keep solving the papers, we will keep discussing. I have not gone into much detail because I want to cover the paper, as many papers as possible, and if possible you can go through the explanation by yourself because it would take time. My purpose is to solve the number of papers with you, all botany papers related to different exams. So because the questions are common, so if you keep on revising from different papers from different exams, the questions are similar, okay. Whatever exams you are targeting, you will solve the previous years of that exam, that's for sure, but the questions in different exams for the same subject are moreover similar. So we will try to do that with you. I hope this has helped. Please like and subscribe the channel, it helps to uh, give us the motivation to make more videos like this. So this all effort is for you all. Thank you so much. See you.